today's video, we're covering liquid biopsies. We're going to look at what liquid biopsies are, some different things that you can look at with liquid biopsies, and finally we're going to look at how the analysis of the liquid biopsy is done. Liquid biopsies are just like a normal biopsy, a test to decide if there are cancer cells present or not. In the blood, there are these biomarkers that are associated with cancer that can be present. A tumor cell will secrete these biomarkers into the bloodstream that in theory could be detected using a normal blood test or urine test, for example. So the blood is being taken up with the present biomarkers for the tumor cells, and it can then be analyzed using different types of methods that we'll take a look at later. And you can then see these biomarkers that are present in the blood for this example. And in the future, many scientists believe that this is the next big thing when it comes to detecting cancer at an early stage. And the biomarkers that you can see are, for example, DNA from the tumor cells, tumor-specific methylation patterns, tumor proteins, as well as tumor-educated platelet cells. Now, before we look at how these biopsies are analyzed, let's take a quick look at what tumor-educated platelet cells are. And if you already know what it is, you can skip ahead to. Now, tumor education is basically the process where the tumor cells reprogram the platelets to work in their favor. The tumor sends out RNA that the platelet will pick up and then be reprogrammed molecules and proteins that are favorable for the tumor. For example, it will express molecules that stimulate the growth of blood vessels to the tumor. They will supply it with nutrients to make it grow bigger. Now, finally, let's take a look at how these liquid biopsies are analyzed. The most modern way is to use something called FTIR spectrometry. It works by exposing the molecules in the sample with infrared light, and this will make the molecules start to shake or rotate. Now, depending on the different molecules within the cells, for example, they will shake or vibrate differently, and this is what is used to decide what specific types of molecules are present in the sample. So when we expose our sample with the infrared light, the molecules within the sample will reflect some of the light, and some of the light will be absorbed by the molecules, causing them to start to shake, and a different wavelength of the infrared light will pass through it. And it's this difference in wavelengths that is detected by the detector. And because different molecules will take up different wavelengths from the infrared light, you can get an image of what type of molecules are present. And the computer will then create these detailed maps that, combined with AI technology, can give a detailed picture of how much tumor biomarkers are present, for example, or inflammatory proteins and other types of cells. It could basically, in the future, give a detailed picture of the microenvironment that the tumors create, making it possible to detect the tumor at an early stage.
Thanks for watching. Please like the video and subscribe.